Good morning once again. I wanted to share some of the kind of interdisciplinary approach about the background and the genetics of sickle cell anemia. So we'll begin by stating that sickle cell anemia disease has impacted the United States and the world both socially, politically, and medically. Sickle cell anemia was once described in this country as a Negro disease because it predominantly affected people of color, people of African descent, and that was a query of people didn't understand why it was affecting only one of the racial groups or ethnic groups in the country. It was also labeled as a form of black genocide, so it was because the mechanism, molecular and medical mechanism, wasn't known at one time. People believed it was a form of black genocide, a way to eradicate the black race. And by self-empowerment in the 1970s and 71 in particular, as you see on the screen, the Black Panther Party began establishing the research foundations in order to try to elucidate what is the cause of sickle cell anemia. So both socially, politically, and medically, this disease has affected America and the world. So it's fitting that we're taking this approach to take a look at this disease. So if we look at just a brief history, why has it affected these particular areas, or these particular ethnic groups? So sickle cell disease is linked with the disease malaria, and it was most prevalent in parts of the world where malaria was present. And we can see that in parts of Africa, the Middle East, Southern Asia, and Southern Europe. We had the presence of malaria was there, as well as individuals were suffering from sickle cell disorder or sickle cell disease. However, since the abundance of individuals who were suffering with the disease were from Africa, the continent of Africa, it became known as a black disease or Negro disease, which was the term that was used at that particular time rather than African American. Of course, so what is the tie to malaria, or what is malaria? So malaria is a disease caused by a parasite that's a protozoan, and it's called Plasmodium. So this parasite is transmitted by the Anopheles mosquito. So the mosquito, when it bites someone, if that person is affected with the parasite, when the mosquito sucks the blood of that individual, it takes the parasite as well. When it bites the next individual, it injects that parasite into the next individual. So the life cycle of the organism lives inside of the mosquito, but once it gets inside of the human body, it goes inside of the red blood cell where it reproduces itself, and then it can burst out of that cell and infect other red blood cells, causing anemia because you're destroying the red blood cells, and a shortage of red blood cells is referred to as anemia. So that's the connection that it has to the disease malaria, because human red blood cells is a part of the life cycle of the organism or the parasite. So how is malaria linked to sickle cell disease? The ability of human red blood cells to host Plasmodium infection. So whether or not the red blood cell has the ability to host that organism, so once the individual is bitten by the mosquito carrying that parasite, can the red blood cell hold that parasite in order for it to do its reproduction and then infect other cells? So that's going to be the key when we begin to look at sickle cell disease itself. So what is the genetics, since I'm here to talk about the genetics of sickle cell? The genetics of sickle cell disease is referred to as an autosomal recessive. So without going too much into the genetics, autosome is, or chromosomes, or description of chromosomes, and recessive means that the trait will skip generations. It's not present in every generation. Pretty much as we would see someone has freckles in the family and it skips a generation and the grandkids have freckles, right? Okay. When we look at a gene has what we call two members, and those members are called alleles. For sickle cell, they are designated as the HB. HB is hemoglobin, which is the protein that's found in red blood cells. If it's normal, it's referred to as A, hemoglobin A. And for the sickle cell, it's designated as S, so the hemoglobin S. And we would say that's the recessive form of the disease. So we have the two alleles of the individuals. have to get a couple of terms before we go to the next slide and look at how it's inherited. Okay. So we have two terms, what we call homozygous and heterozygous. The prefix homo means the same. So the two members of that gene pair or the alleles are the same. Or if it's heterozygous, hetero refers to different. And so the two members of the gene pair are different from one another. And so I've provided examples of those for you here. So the two alleles we have, the dominant is the HBA, whereas the recessive is the HBS. A homozygous individual who is normal would be HBA. The two alleles are the same or identical to one another. For a person who has sickle cell disease, they would have the two HBS alleles. For a person who has sickle cell trait, as we call it, they have one sickle cell allele and one normal allele. We refer to them as being heterozygous. And we'll talk about those, and particularly Nurse Robichelle will tell us a little bit more about sickle cell trait when she's up. Richard will tell us a little bit more about sickle cell trait when she's up. So sickle cell actually follows an unusual pattern of heredity. Many of you probably have heard of Gregory Mendel and his garden peas, and he's the father of genetics. Well, Mendel couldn't explain, or Mendel's laws and rules do not explain the pattern of heredity for sickle cell disease. And so we call it an extension to Mendelian inheritance. And this form of inheritance is referred to as overdominance. Overdominance. An older term for it was called heterozygote advantage. And so we want to see how does this apply to sickle cell anemia. So individuals, I showed you homozygous and heterozygous. Okay, so for individuals who are heterozygous, having the one normal allele and one recessive allele, their red blood cells, if they're infected with a parasite, the cell will burst open, and therefore the parasite would not be able to continue its life cycle. Therefore, they would not become, all the red blood cells would not be infected, and this person would never get anemia. So it was an advantage for the individuals to have the heterozygous form. If we look at the two homozygous forms, the HBS, these people suffer from sickle cell disease. Before medical intervention in our current Western medicine, these individuals died before their reproductive age. So individuals who had sickle cell died before they were 12 years old. So it's that devastating of a disease. So individuals who were living in Africa, Southern Asia, Southern Europe, all of those, the children were dying before they reached the age of 12 years old if they were affected by sickle cell disease. Individuals who were HBA, they had no trace of the disease at all. However, they were susceptible to malaria. So they were living in the same areas. The mosquitoes were biting them, injecting the parasite to carry out its life cycle. So those people were dying of malaria. The homozygous sickle cell were dying of sickle cell, and it was the heterozygous individuals who were alive and being able to be resistant to the sickle cell disease. These were many of the people who then were captured and brought over in the Middle Passage, and that's how we African Americans have the sickle cell trait coming from our ancestors who were brought over in the Middle Passage to America from Africa. What are some of the physiological effects of sickle cell disease? So individuals have red blood cells that deform into a sickle shape, and I'll show you what a sickle shape looks like. It's like a crescent moon. We use the crescent moon oftentimes here in New Orleans to represent the city. And so our cells, which are supposed to be a round disc, actually change the format into a sickle. Okay? What are the two ramifications of that? Some of the ramifications of this is the sickling greatly shortens the lifespan of the red blood cells. So the red blood cells reproduce or regenerate themselves throughout the life of the individual. These red blood cells have a shorter lifespan, therefore you have fewer of those red blood cells, and that's what the anemia is, a shortage of red blood cells. The odd shape of the red blood cells causes them to clog in the vessels, the blood vessels, and that's going to lead to pain to individuals, a decrease in the ability to carry oxygen, because that's what our red blood cells do, carry oxygen and supply that to the brain. Okay. And in may cause the individual to have an overall shorter lifespan. As we saw, mentioned earlier, individuals before medical intervention died by the age of 12, but now with lots of medical intervention, we have individuals who live well into their 40s. I think one of the, T-Boss from TLC is a spokesperson for sickle cell anemia, and she's lived with the disease for quite some time, and she's well into her 40s, I think getting close to 50, if not already there. So the lifespan of individuals with sickle cell anemia has increased dramatically based upon medical intervention. One of the other things we want to do, because a common question becomes, what are the chances that I will pass the traits on to my children, or what's the probability that it runs in my family? So as a geneticist, this is what we do. We usually would draw a Punnett square, but instead I drew a little picture, or inserted a picture for you there. So what if you have two people, a couple here, who have sickle cell trait? That means that both of them are heterozygous. They have one normal allele and one sickle cell allele. Okay? So they marry and they have children. Okay? So one allele from each parent goes to a child. So if dad gives his first son the normal allele with the A, and mom gives him also her normal allele, then this child has no chance of ever acquiring sickle cell, and he will not pass it on to his children because he doesn't have the allele to pass. The two children in the middle, suppose in the first son here, he received a normal allele from his dad, the HBA, but he received the HBS allele from his mom. So he's heterozygous, he carries the trait. He does not have the disease, but he's able to pass that trait on to his children when he has children. The same with the third son. He received the opposite, the recessive allele from his dad, the dominant allele from his mom, and therefore he's heterozygous. The last child on the end received the recessive from his dad, the recessive from his mom, and therefore he has sickle cell disease. So we are able to calculate or to talk to a family as a geneticist. If two people carry the trait, we would tell them you have a 1/4 chance of having a child who has sickle cell disease. And that can then come into play if they are planning their family and deciding whether they want to have children or not. 1/4 of their children will be completely normal, so you have the same probability. Half of the children will be carriers of the trait but will not have the disease. So you still have 3/4 of your children who would not have the disease, a 3/4 chance of having a child without the disease, and only 1/4 chance of having a child who actually suffers from sickle cell disease. In order to know whether or not we carry the alleles, we have to have a genetic test done, and they will run our DNA and to be able to tell you whether or not you have the disorder. That's usually not covered under your insurance, so that would be something that you would have to do for a DNA test as compared to the medical test, which maybe talked about Dr. Richard, where babies are tested for the presence of sickle cell on birth. And this one, on the molecular level, we begin to look at the resistance of the disease based upon the allele formation, and then how is the protein formed. So we'll just move forward on that one and just show you a picture. So what happens with sickle cell disease? How did it come about? It comes about by a mutation, and a mutation is a change in the DNA. So way back in history, back in early human existence, there was one change in the gene that encodes the hemoglobin protein. If you look at top, there is the normal, and it says GAG-CTC. If you look at the middle nucleotide, so this is representing a short segment of our DNA. In the mutated form, where you have an A normal, you have a T in the mutant form. Okay? So there's one change, one letter, one nucleotide has been changed in the entire molecule of DNA and the entire gene. Genes are being translated to make our proteins, and so we get a different form of the protein. If we look at the fourth box of that, the colored box, at top we have a GLY for glycine, whereas in the mutated form you have a yellow box with VAL, valine. So you have a different amino acid produced, put into the protein based upon that one change in your DNA. How does that affect us? The shape of the protein is different. In the normal, you have hemoglobin molecules individually. But in the sickle cell, they become rope-like, actually giving us that sickle shape. Okay? So the protein shapes very differently. So based upon the amino acids that are in the protein, it determines the shape. The shape determines the function, whether it functions properly or not. Okay? So that's what we want you to see. So one change in the DNA causes this particular disease. What's happening inside the cell? Here we have the two shapes of the cell. We see that the blood flows normally, and the other individuals will talk about the pain that's caused by the clogging up of the vessels when you have the sickle cells there. And so that's on the bottom. Cure right now from the NIH has initiatives right now. There's bone marrow transfusions. So you're often having blood drives or bone marrow drives in order to work towards a cure for sickle cell. And also right now on the horizon is gene modification or gene therapy using the process called CRISPR, which is a gene editing process where we'll be able to go back and change that one nucleotide that was wrong. This mechanism can go and change it back to the correct one and be able to get rid of the disease. And they're doing that in those cells in the bone marrow. So that's it. Here's the invitation to my talk on next week. It's going to be in the library at 7 p.m. And I'll be speaking from my book there. And so I'll take questions from you when it's my turn on the panel. Thank you for your attention.